I've been wanting to do this story, Tatum, for like 3 or 4 weeks now. This is one of the coolest studies, I think, of the year. That's big praise. It's a fish study, Tatum. This study is a means by which that there is a genetic shift in parental care. So it is taking an animal, a cichlid fish, where females have parental care and males don't. It is doing some genetic tricks and having the males have and perform parental care. So this is the story that I was teasing a little bit ago about deadbeat dads, as it were. These fish are just rude dads. They're just like, they're papas. They're like, I don't want to have anything to do with my children. Goodbye. The way this study started was in the form of panic. I think this is really cool of how this story started because, as Jay asked with like the new species, oh, did they intentionally go out and look for these new species? Was there a goal here? Right? Like, in seeing this paper, right, was their original goal to identify what gene controls parental care and if they could make that switch? Like, was it a very educated, well thought out plan, or was it a total accident? So, Jay, it turns out this was a total accident. So normally the female cichlid fish, they have a protruding jaw. Their mouth is like popping outward a little bit, and that's signifying that they're carrying embryos. And the image that you can see is right here. So they have that lower little almost sac there is sticking out, and they are holding embryos in their mouth, and it's a means of protecting the individuals that are in their mouths. It's incubating the eggs and keeping them safe until they're big enough to swim solo. So even after hatching, when the fish are really tiny, they'll hang out in mom's mouth until they're ready to go out. However, Jay, researchers in the University of Maryland were hanging out, and there was a postdoc to a researcher, and he looked in the tank and he saw a fish with its mouth extended, like that lower part. And you're right, normally, Jay, that's not a big deal. Like we just said, all the ladies do it. But it was this guy. It was a male, Jay. It was a male, and it was extending the underside of its mouth, and it was holding embryos. So this was a natural member of the population holding these embryos, male. And the researcher was like, oh God, I've made a mistake. I've sexed this fish incorrectly because there's just absolutely no way, right? No, he had sexed it correctly. It was a male caring for the embryos where it never should have, because it is never otherwise seen. Largemouth bass, the males care for the little ones after they hatch, and the mama bass goes on. Really? That's cool, Tatum. So I guess, Tatum, it would be also cool then. So we're going to talk about what the genetic side of this is. I wonder, Tatum, if the genetic answer found in this fish is the same as in the bass, except it's just reversed, right? Like, the genetics that prevent this male from carrying, right? Maybe it's just that gene is on in the opposite direction in the largemouth bass, and that's why that they take care of their offspring versus these don't. As a possibility. That, right, I don't know, but that would be really interesting to see how well this is conserved. So Jay, but why it's cool, right? If it's never ever seen, and he looks inside and he sees it, he's like, well, what on earth is this? And so it's actually, the way they had an interview on it. It was like, this is the first male that he'd ever seen do this kind of mouth breathing where they're holding the embryos. And so what they ended up doing was they put in a bunch of cameras. And they were just like, all right, we got to see if there's other males that do this too. And they found, Jay, a whole batch of males in the tanks, in one of the tanks that was doing this behavior. They're like, okay, something's off with these males. They did a genetic sequencing. They had to do full genome sequencing now. So this is not like with the new species of the week where we just do a little sequencing, right? And it's like, no, full genome sequencing. Yeah, so Andrea, what this meant was there was a mutation in one tank that spread, and this one mutation was responsible for this behavioral change. And before you do the sequencing, you don't know how many mutations there are that are causing this behavior. But what was really interesting was it was a specific mutation. It was one mutation here. So how did so yeah, exactly. So Jay, they would have sequenced a couple of fish. They would have taken the reference genome. So the genome of this fish was already sequenced before. That is called the reference genome. That is what you compare against. That is your, quote, normal. And then you compare the genome sequence against that normal of the mutants and see what is different. And so the difference was this gene. They did, oh yeah, werewolf's like, Balint, if you find this cool, and it might be kind of cool, you gotta replicate the mutation, man. You can't just be like, oh, we sequenced a mutant and we found something similar. No, they did werewolf. They did do the replication here as well. And so it was one gene that they found. Now, normally, it takes, like, if we look at the biology of this fish, and again, I have all these notes written out on the fish biology because I knew Tatum was going to ask about the fish. And so I wanted to know about these fish. So Tatum, these cichlids take about 2 weeks from going to egg to free-swimming juveniles, meaning that they will be out of there will no longer be mouth brooding, like within the parents. 2 weeks. During that 2-week period, the fish carrying the embryos cannot eat because they're holding in the embryos. So, Jay, no lasers in this one, but they did find the gene. And actually, here's where it gets wild. It's a pheromone receptor. It is a smell-detecting gene called OR113A. OR meaning odorant receptor, and then it's the numerical Q afterward. So what they think is going on is that there is detection by the male of a female pheromone, and it prevents that circuit from being activated. So it's an inhibitory smell that makes the males be the deadbeat dads. And as soon as you get rid of the ability of the male to smell that, the male takes care of the developing embryos. Does that make sense of the direction? So essentially, when the males come close and it's time to lay the eggs after mating, the smell is released by the females, and it causes the males to stay away from the eggs and avoid getting them. The hypothesis is why this exists is that maybe, like, some species, males will actually eat the fish, and so this is a way to avoid the fish from being eaten. So, right, it's a crazy circuit and how it works of this fish. And again, they found this one mutation between the two fish, and then they got to Werewolf Cass's question. They were like, all right, Werewolf is gonna ask us to replicate this. Werewolf is the reviewer of this paper. Werewolf was like, all right, you did some mutation sequencing, that's fine. But until you can replicate this in a wild type normal fish by mutating that one site, I don't buy it. Werewolf, you are a very good reviewer. You are a top tier reviewer, Werewolf Cass. And so they did it. The reason this is a big deal is they use CRISPR genetic engineering to get rid of a gene in this animal. So they got the wild type normal fish, right, where the males don't take care of the embryos and the females do. They went in and they changed that one gene again, Werewolf. And they changed it by shutting down that gene, that odorant receptor. Why this is a big deal is because every time you do CRISPR, every time you do genetic engineering, and you're turning off a gene in an animal, it sounds straightforward. It sounds so straightforward, right? It's you insert a guide sequence, and the guide sequence binds to the DNA target of your interest, and then you throw in some Cas9, and Cas9 binds to the double-stranded DNA that you've targeted, and it chops and it breaks it, and then you've introduced a mutation. It sounds so easy. Every single animal that you inject and you try to do this experiment on has to be optimized every single time. The right time to do the injections during development, the concentration of them, which if you need a different kind of Cas9 to do it, if you need to insert something, it is not straightforward and simple, but they went ahead and did it in this paper. So the sequence difference is there are presumably other variations in the genome as well. So there are what are called non-coding differences in the genome. So there's genes that make proteins, right? And there's genes that don't make proteins, but they influence the structure of the DNA. So because they don't they don't code protein, they're not always looked at in terms of the how is it said, like the mutation load. But what that means is that there still could be some off-target effect, meaning that there could be a mutation elsewhere in the genome, even though you identified the difference between this fish and this fish is this odorant receptor is very different. there are other mutations scattered along in there that are just maybe differences between individuals, right? Like, let's say, Jay, you and I are being compared, our genomes, my genome to your genome, but you have wings. And I find a gene or two that looks like is what gave you wings. Are you satisfied with me saying, all right, it's those one or two genes that gave you wings? Or would you want to look at and do an experiment where you were the one that actually changed those two genes and nothing else, and then you were able to demonstrate if there was wings or not? So basically what you'd be trying to do is replicating it in a genetic background that's the same. So if we take the fish that's doing the normal brood care, right, where it's only the females taking care of the embryos, the males don't, everyone in that tank, Jay, is genetically identical. So this is a important element of the sciences. When you're doing these kinds of experiments, you have to know the background genetics of the animal that you're testing. So with fruit flies, every fly in a vial is genetically identical. So if I test 2 flies within that vial, it should give me the same result every time. If I take a different vial of flies that is a totally different background, meaning like it's still normal, but if you were to compare the two genomes, they'd be different. Well, what are you calling, which is normal? So here you take something that you're defining as normal, and you are altering the one gene in there and seeing what ends up happening. Does that make sense, Jay? So again, you're comparing your wild type, your one that you know is behaviorally normal, and you yourself are changing one gene. And by changing that one gene, you're hopefully recapitulating the same behavior. So it's a term in genetics called necessity and sufficiency. You want to be able to demonstrate that the genetic change that you're observing that you introduce is necessary to the behavioral shift, and it's also enough to cause the behavioral shift. So what I mean by that is the only extra experiment that they could have done to truly convince me of this is if they took that mutant fish that they identified in the tank and they reverted it. If they fixed that gene in the mutant fish and made the dads not care again, then I would say, okay, you've done both elements to it. But here they just showed necessity, where if you get rid of this gene, the fathers now will start caring for the embryos. Let me know, y'all, if that makes sense or not. Hopefully that's making some semblance of sense. But yeah, I think it's a really, really, really cool experiment that they ended up doing. So again, when they turned off that gene using genetic engineering, they found that it wasn't even like a total number. And maybe, Jay, this also gets to kind of your point too. Let's see, makes sense, but how confident do you need to be in the gene sequences to go the extra step to replicate another experiment? So what's really neat is that they had a graphical abstract in this paper, and I wish more papers would have graphical abstracts because it's a nice visual cue of like, what did they actually find in this paper? And so again, remember, as we said in the takeaway was that there was a olfactory receptor, this OR11A3, that caused there to be this lack of signal to be detected. And usually the signal is detected by a male from the female, and that inhibits a parental circuit. However, if you take away the inhibitor, the normal state is for the parental circuit to be on. And so this is what the imagery that you're seeing here. You've got these pheromones coming from the female, and that inhibits the egg retrieval or parental care on these animals. But if you take out this olfactory gene, it goes in the other direction. So this is the normal again. The males are at first attracted to the females. There's mating. Then only the females take care of the embryos by retrieving them and actually hold them in their mouth. And then if you do this mutation, and actually they found a second mutation that did the same thing, there is actually a reduced attraction, suggesting that this olfactory sensor is in part implicated in the original courtship behavior. But then, again, what's really interesting is both of them, male and female, end up collecting the embryos. Hopefully that makes sense, y'all. Again, I think this is a phenomenal paper because it's looking at parental care behavior. It was identified from a random mutation within the population that was being kept within the lab, and then recapitulated in a genetically controlled manner using CRISPR genetic engineering. Like, this is not an easy and straightforward experiment, and yet they did it. So I was, it was like my hat's off to y'all. But yeah, here are some of the elements that they're looking at the fish, like giving them the fish the different kinds of odorants as well, and trying to plot the position of the animal, of how they move and where they move to as a response to these odorants. What they actually did that I thought was really cool was they did single cell sequencing. So within the brain, you can either do a bulk genetic sequencing or you can do sequencing on a single cell level. What's really cool about this is if you do it on a single cell level, you get this kind of plot here. This is called the Tisney plot, where each cell group corresponds to something different if you've done the experiment correctly. So here, this cluster is a certain kind of immune cell. This cluster is the olfactory sensory neurons. That's the one where they're going to be looking at. That's the most important one, like for this relevant experiment. There's immune cells, there's hair growth, etc. that you're being able to see here, like looking at the single cell map and resolution. And then what you do is pretty simple, is that you do mutations and you look at comparisons, see like, all right, what is different from the big graph and these smaller graphs where you introduce the mutation? So here are the actual fish. I thought, again, this paper was really remarkable. They made the genetic mutations using CRISPR in these fish. And again, you can even see there's like this extension here in the mouth structure where they're going to be keeping the embryos right down here versus, so this is your normal fish, this is a mutant, this is a normal gene set, so ++ means normal, triangle or delta means that you've removed that gene of interest and now you've caused that mutation to be present. That's another element to it that I think is really cool. And then here's a wild type female. So they looked at the different genetic elements here. They've also then tested them for their behaviors. But yeah, so here we go. Let's see which data set. Yeah, I mean, they looked at the mating differences, which was interesting. Like there's the differences in them. There's a lot going on here in the figures. But like, again, really, really nicely done. Preference indices, we've kind of gone over these elements as well. Here we go. This is the big data set too. Is that when upon mutations, I thought this was a neat little trick that they did as well. Having a single mutation, so making it a heterozygous in the fish. So one copy of the mutation and one wild type copy was not enough to cause any male egg retrieval. However, if you had both genes mutated, it was enough to cause some of this retrieval. Not all the genes they mutated caused this. They found the second element, the CNGA2B mutation, that did induce sometimes the male egg retrieval, about 3% of the time, better than 0. But the olfactory mutation was about 30 to 40% of the time it induced male egg retrieval. This also suggests, y'all, because it was not 100% of the time that the males were picking out the embryos, because it wasn't 100%, there must be other genes involved as well. The researchers identified one major component of this olfactory gene that is responsible for a big chunk of the behavioral preference and behavioral change, but not entirely responsible for it. And again, what's really cool is not only does this gene involved in the egg retrieval behavior, but it's also involved in the mating behavior, the approaching behavior as well, like when starting off. So there's some kind of very complicated circuitry going on here where it's important to have that allows the animal to be detected, like in terms of the mating origination, and then they keep on this olfactory receptor, so when the male then smells the female after eggs are laid, a particular odorant that comes off the female, it makes the male go away and not be involved with the brood care or taking care of the embryos. How often is it that a single gene affects behavior in anything? So for this one, Smikes, it's probably depending on what kind of gene that you're getting rid of. So if it's an upstream regulatory gene, meaning it's a gene that regulates a lot of different components, if you take that out, then it might be this single gene you found controls the behavior. And I've seen some people pitch that. My argument is that is not a single gene paper. That would still be you didn't identify the gene, right? You identified a regulator that controls a ton of downstream genes and you don't know which one actually matters. In your questions, Mike, if you actually get to the single gene affecting that behavior, it's very rare that it's going to be that single gene. So it makes sense here, right, too, that with this olfactory receptor, maybe it's a big chunk of the behavioral change that you're seeing, but it's not the whole story. And again, given the complexity of a living animal, this huge parental change is probably, it makes sense it's only a single one. All right. And then this, I thought, again, it was a neat study too, where you're looking at, here's the wild type, the olfactory receptor, and then here's the mutant, and they're analyzing time that the males spent retrieving the eggs, and the red is that mutant, and almost all the time is spent now retrieving the eggs, versus the wild type, like, they're not gonna be messing with the embryos. So I think it's a very convincing study. I think the reason that it wasn't a higher tier paper is because they only showed necessity and not sufficiency. What I mean by that is, they removed the gene in a normal fish, and when they removed the normal fish gene, they were able to introduce this behavior of male parental care, which is otherwise never seen. What they, I think, should have done was go into the original mutant fish they found. So Volcanodoc, I don't know if you heard the beginning of the story, but these fish never do this parental care. And the postdoc doing the work went into the lab one day, and in one of the boxes, right, they found male fish that were doing this brood care. And so they did the sequencing where they got the candidates, and then they took those candidate genes and knocked them out in a normal fish, and then got this result. Now, I think the final thing what I've been doing is, sufficiency. So it needs to be necessary and sufficient. So the sufficiency element here, what is coming in on that stage is what they should have done, the final step, is to take that mutant that was naturally occurring in the lab and fix the gene. If they fixed the OR113A gene, restored it to its normal function, and then the males no longer cared about the babies, that would have been, okay, this gene is necessary for the behavior, and it's sufficient for it too. And that's getting at both sides of the story. Here, they analyzed one side of it really, really well. But if they would have done that additional step, that might have been enough to get into a higher tier. I don't know for sure, Smikes, if that would have gotten them in to that higher tier. Usually Cell Science and Nature want also some kind of statement of why this is important for human health, or something very sexy and flashy. There's not really anything like that in this paper beyond it's just really cool science. I think they could have framed the argument maybe a little bit differently of why parental care is really cool. And there is something confusing here that I have yet to point out. The confusing thing here is that because it's only one gene, that kind of makes it almost confusing as to why it's a single gene. And again, by that I just mean, like, if one mutation can cause the dads to take care of the embryos and to be less receptive to females, that seems like it's a system that's ready to be broken quite easily. So I don't know, there wasn't really any addressal of that. With, again, just one gene being such a big component of this, it does seem a little strange. But yeah, it's still interesting. I don't think it's just the money, to be honest with you, Miss Goinian. I think it's just difficult. What I think that's going on here is that maybe they did try to restore that gene, and it's just, as Mike said, it's not a single gene. And so maybe there are genetic effects sometimes of knocking out a gene has a really big effect. Like here, 35 to 40% of the males would now do brood care versus 0 did it before. Restoring this one gene maybe only change the effect by 5% because there's something else in there that is also causing there to be a big effect, right? Because they didn't test all of their candidates. There may be another mutation in that candidate list that's more important. They went for one that was identifiable and easy versus testing like 30 different mutations. And presumably part of that is just because it's difficult, because they had to address and make genetic engineering tools in an animal that didn't have it already. That would be my guesses of that. So it's cool to see because, right, they keep the embryos in her mouth for 2 weeks, and so all these adult, like, little mini fish are coming out, meant that the adult was unable to eat during that time. What's interesting here, y'all, and they didn't look at this in the paper, is what happens after the offspring hatch. Like, is there swimming around following the male? Does the male continue some kind of, like, protective behavior? That they didn't look into. They just looked at carrying the embryos in the male's mouth. So we don't know the full story, but I think that'll be like a neat next experimental step that they can do. So yeah, it's only 40%, and it makes sense too that it's not solely on one gene, because if it were, you can imagine a situation where if that one gene mutated and it caused so many effects in the wild, there would be no buffer to their natural behavior, which may or may not be a bad thing. Researchers didn't in this paper demonstrate what the negative effects were of male parental care. Like, what is the downside to it? Why isn't this the normal state of things? It was just trying to identify what is the genetics behind this behavior, how does it work, and why does it work in this way. Well, so Fishman, what was funny, at least to me, is that the popular media articles framed this whole thing as, don't be a deadbeat dad, or fish that are no longer deadbeat dads, or how to correct being a deadbeat dad was like the way that the popular media article was framed. And I was like, what's, so that made, I was like, what is actually going on in here? And it's not quite what's going on, because they aren't just leaving because they're just like, oh, I don't care about these embryos. They're not normally interacting with the embryos because there is this olfactory receptor that they have that when they smell from the female, they leave. Like, it is an inhibitory signal that tells them to go away, right? Which to me is different than being a deadbeat dad. A deadbeat dad just means like, hey, I'm just gonna peace out. I'm just gonna head out right here and just not participate. That's not what these fish are doing. So, I don't know. It's a fun little primer of a news article, but it's not accurate.